Welcome to my channel, Deep Learning Biology. My name is Dr. Vikash Bhardwaj. Now in this video, you are going to learn some of the calculation related to DNA, RNA quantification. So I am going to put few of the videos in which we are going to discuss some of the important calculation related to DNA, RNA quantification. Now here I just want to tell you, you must be knowing, generally we use UV spectrophotometer for quantification of DNA, for quantification of RNA, for quantification of proteins within the laboratory. Now here if you see, in the case of DNA, in the case of RNA, okay, you know very well the base, okay, purines as well as pyrimidines, okay, they are responsible for absorption of light. So within the DNA, within the RNA, you are going to have bases, okay, and these bases generally they absorb UV light at 260 nanometer. So keep in mind, in the case of nucleic acid, the maximum absorbance of light, it occurs at 260 nanometer. So at this point, the 260 nanometer, okay, at 260 nanometer wavelength, generally we quantify DNA and RNA. Now try to see here, okay, hopefully you must have seen a spectrophotometer within the laboratory, okay. So here what you are going to have, you are going to have a cuvette, okay. Now here in case of nucleic acid, we are going to use quartz cuvette, okay. Why? Because quartz, it is not going to absorb any UV light, okay. So here you are going to have your initial, what you can say, bombardment of UV light, okay. So from here the UV light will come and it will pass through the quartz cuvette, okay, and within the quartz cuvette you are going to have your sample, okay, and what amount of light it is going to absorb, that you will calculate, that you will see with the help of spectrophotometer. Now try to see here. Here let's suppose initially, okay, generally in the lab you must have seen what we are going to have. Initially in the quartz cuvette we are going to have just buffer, okay, a blank solution, okay, without any DNA, without any RNA. Now here, so the UV light, okay, generally we are going to use, and the wavelength is 260 nanometer, okay. Light will pass through the buffer, okay. Let's suppose it is going to absorb X amount of light, okay. Now there can be another situation. In another situation, what you are going to do, try to see, you are going to have the sample buffer now with your DNA, okay. Same way here also you are going to use the 260 nanometer wavelength of light, okay. Light will pass through the cuvette, okay. Now let's suppose in this condition Y amount of light it is being absorbed. Try to see here your buffer, okay, it is going to absorb X amount of light. Your buffer plus DNA, it is going to absorb Y amount of light. What amount of light will be absorbed by the DNA? It is y minus x. Y minus x. Okay, so this way we can calculate easily what amount of light was being absorbed by DNA. Now, you know very well, most of the times nowadays we are going to have spectrophotometers. Okay, and generally what we do nowadays, okay, we will prepare one blank. Okay, remember this blank is going to have just only the buffer. Okay, here we are going to use 260 nanometer wavelength light. Okay, this light will pass through the quartz cuvette. Okay, now the light will be absorbed by the buffer also, but here we will program our spectrophotometer in such a way that the spectrophotometer will consider this light as 0. We will just blank this. Okay, now next here we are going to have our sample. Okay, in this sample we are going to have buffer plus DNA. Okay, now here what amount of light will be absorbed? Okay, it will be seen as absorbance. Let's suppose it is 0.53. Okay, so here you see now the spectrophotometer, it can directly tell you what is going to be the absorbance. Okay, so here this way we calculate the absorbance. First we are going to blank the instrument. Okay, so whatever the amount of light the buffer is going to absorb, it will consider 0. And now whatever the amount of light DNA is going to absorb, it will show you. Okay, so hopefully it is clear. Now keep in mind that in case of DNA, okay, we are going to calculate absorbance at 260 nanometer. So this is for DNA. This is also for RNA. Okay, so remember DNA, RNA quantification, whether it is double stranded DNA, whether it is single stranded DNA, whether it is single stranded RNA. So in case of nucleic acid, we are going to do quantification at 260 nanometer. Remember, when we are going to quantify proteins, generally we take the UV light, we calculate absorbance at 280 nanometer. Okay, so here by doing quantification at 260 nanometer, okay, we can easily quantitate what amount of DNA or what amount of RNA is present in our sample. Same way, by performing quantification at 280 nanometer, we can easily find what amount of protein is present within our sample. So here 260 nanometer for DNA, RNA, 280 nanometer for proteins. Okay, now here keep in mind, okay, so if your sample of DNA, okay, it is pure, okay, generally we are going to calculate for pure DNA. Okay, suddenly we calculate this ratio A260 by A280. If this ratio comes out 1.8, okay, then you can say the DNA is pure enough. Okay, it is pure. But sometimes you can have reading greater than 2. Don't think that the sample is pure. Okay, it may have some contamination of organic solvent. Okay, and if this ratio is less, okay, less than, you can say 1.5, okay, it is lesser, okay, then you can say your sample is not having pure DNA, it is having contamination of proteins. So here keep in mind, most of the times when we are going to work in the laboratory, we also check this absorbance 260 by absorbance 280 ratio. Okay, so hopefully these basic points are clear to you. Now here I just want to tell you some of the relationships you must remember. Okay, so here when we are going to calculate for double stranded DNA, keep in mind, okay, 1 OD object, okay, absorbance at 260 nanometer, it is equal to 50 microgram per ml. For single stranded DNA, okay, 1 OD, okay, it is equal to 33 microgram per ml. And for single stranded RNA, keep in mind 1 OD, it is equal to 40 microgram per ml. Okay, these points keep in mind. For double stranded DNA, absorbance at 260 nanometer, okay, one absorbance, okay, 1 OD value is equal to 50 microgram per ml. For single stranded DNA, 1 OD is equal to 33 microgram per ml. For single stranded RNA, 1 OD is equal to 40 microgram per ml. Everything will become clear if you are going to solve some of the numerical problems. Okay, let us try to solve some simple numerical problem. Now try to see here. Question: a double stranded DNA solution has absorbance 260 value 0.5. okay, what is the DNA concentration in microgram per ml? Millimolar. Now here what they have mentioned, a double stranded DNA solution, absorbance 260 value 0.5. what is the DNA concentration? And you have to give the answer in microgram per ml as well as in millimolar. Now try to recall 1 OD, okay, it is equal to 50 microgram per ml for double stranded DNA. Okay, so here after spectrophotometric analysis, if you are going to get the value 0.5, okay, 0.5 OD, it will be 50 into 0.5. okay, if you solve this, your answer will come 25 microgram per ml. So here, okay, the sample, okay, which you have quantitated, okay, and if this sample is showing you 0.5, okay, OD, okay, so the value will be, so here the concentration will be 25 microgram per ml. Now to express the answer in millimolar, okay, so here you have to keep in mind, okay, so molar extension coefficient, hopefully you remember this Lambert-Spears law, absorbance is equal to epsilon C into L. So here molar extension coefficient for 1 millimolar double stranded DNA solution is 6.7. Keep in mind, for double stranded DNA, okay, which is going to have a concentration 1 millimolar, the molar extension coefficient value is 6.7. What does it mean? It means 6.7 OD, 6.7 OD, it is equal to 1 millimolar double stranded DNA. Okay, 6.7 OD, 1 millimolar double stranded DNA. Okay, now here you have to calculate, okay, what is going to be the answer in millimolar. Okay, 6.7, 1 millimolar double stranded DNA. Okay, then 1 OD, 1 by 6.7. And what will be 0.5 OD? Very simple, 1 divided by 6.7 into 0.5. Okay, so if you are going to solve this, okay, hopefully the answer will come 0.075. Please check, okay, do it yourself. Okay, so this is the answer in millimolar. Quite easy, just you have to keep in mind 6.7 OD, it is equal to 1 millimolar double stranded DNA. Okay, now let us move forward, okay, and try to solve another numerical problem. Very simple. Okay, question: a solution of DNA has concentration 0.03 millimolar. What is the concentration expressed in picomole per microliter? A solution of DNA, here also you are going to have double stranded DNA, has concentration 0.03 millimolar. What is the concentration expressed in picomoles per microliter? Now here I want to teach such kind of question, okay, because sometimes you have to express the answer, okay, and sometimes students they find difficulty, and these questions are very simple. Okay, now try to see here. Molarity, it is equal to numbers of moles dissolved per liter. Now here your solution is 0.03 millimolar. What that means? Means you have dissolved 0.03 millimoles per liter. Okay, now here you see, if you are going to have 1 molar, okay, means it is going to have 10 to power 3 millimolar. Okay, 10 to power 3 millimole. And if you are going to see, if you want to convert 1 mole into picomole, it is going to be 10 to power 12 picomoles. Okay, now try to see here. Here if you have to convert millimole to picomole, okay, there is going to be a difference of 10 to power 9. So here we have to convert millimoles, okay, into picomoles. Okay, so here what you are going to do, you will multiply by 10 to power 9. Okay, so here try to see 10 so you see 1 mole, it is equal to 10 to power 3 millimoles, 10 to power 12 picomoles. And you see, if you have to convert, okay, millimoles to picomoles, okay, you have to multiply it with 10 to power 9. Now here you have to see, okay, try to see your answer. Try to see, here it is 0.03 millimoles per liter. Try to convert this, okay, just multiply it by 10 to power 9. Okay, so 0.03 into 10 to power 9 picomoles. Okay, now here 1 liter is going to have 1000 ml, okay, or you can say 10 to power 6 microliter. Okay, so here you see we have to express, check what was the question, what is the concentration expressed in picomoles per microliter. So here liter is going to have 1 liter is equal to 10 to the power 6 microliter. Now try to see here. Here, if you solve this, okay, 10 to the power 6, 10 to the power 9, okay, you cancel 0.03 into 10 to the power 3 picomoles per microliter. And if you are going to solve this, okay, it will come out 30 picomoles per microliter. So hopefully you have learned this. Okay, thank you. Please do like, share the video. Okay, we are also going to put more videos related to DNA, RNA quantification. Okay.